Friends, we shall discuss about some of the basic biotechnology multiple choice questions here. The first question is, term biotechnology was first coined in the year 1919 and that was coined by Carl Ericke. Biotechnology is used in areas including agriculture, bioremediation, food processing, etc. In agriculture, genetic engineering is being used to produce plants that are resistant to maybe insects, weeds, plant diseases. So here the option is D. That is all the above. Immunoassays are used for drug level testing, detection of unsafe levels of pesticides, herbicides, and toxins on crops and in animal products. Genetic engineering is use of cellular enzymes to manipulate DNA, then improve existing or creating novel proteins to make useful products, then block or increase the production of certain proteins, then grow cells or tissues under laboratory conditions to produce an entire organism or to produce new products. These are the options, but at basic level, the genetic engineering is use of cellular enzymes to manipulate DNA. That is also called as genome editing also. Protein engineers or protein engineering is use of cellular enzymes to manipulate DNA. B option is improve existing or create novel proteins to make useful products. C is block or decrease the production of certain proteins. D option is grow cells or tissues under laboratory condition to produce an entire organism or to produce new product. Here the answer is improving the existing or creating novel proteins to make useful products, that is protein engineering. Antisense or RNA interference technology is options are use of cellular enzymes to manipulate the DNA. This is genetic engineering. Then improving the existing or creating novel proteins to make useful products. This is for protein engineering. Then block or decrease the production of certain proteins. This is the answer for RNAi technology. Ninth one is cell and tissue culture technology is, the answer is grow cells or tissues under laboratory conditions to produce an entire organisms or to produce new products. Friends, I have taken this multiple choice questions from AgriMon's site. Bioinformatics technology is, it is a computational analysis of biological data. I repeat, bioinformatics technology is computational analysis of biological data. Next question is functional genomics. Functional genomics is a high throughput approach to determine the biological function of all the genes and their products. Translational genomics include transcriptomics alone, proteomics alone, metabolomics and transgenomics, all the above. Here translational genomics include all the above, that means transcriptional, proteomic, metabolic and transgenomics. Next is metabolomics includes metabolite profiling, chemical footprinting and flux analysis. Next is various technologies applied in plant biotechnology include genetic engineering, recombinant DNA technology, that is nothing but genetic engineering, then tissue culture and molecular breeding, that is marker assisted selection. All these technologies are included in biotechnology. Vaccination was first attempted by George Mendel, Oswald Overy, MacLeod and Edward Jenner. Vaccination was first attempted by Edward Jenner and that was in the year 1797. Next question is laws of inheritance was discovered by, that is Gregor Mendel, who gave 3 laws of inheritance: law of dominance, law of segregation and law of independent assortment. Science of genetics was born in the year, This was in 1865. Why 1865? Because in 1865, Mendel published his paper in Natural History Society of Brunn, which was published in German language. Okay, and along with that hereditary principles publication, the year 1865 is considered as Science of Genetics born year. Phages infect bacteriophage. They infect only bacteria because phages are kind of viruses only. Okay. Name the scientist who discovered that radiation causes defects in chromosomes. This was given by one in radiation genetics was studied by Hermann Muller. For this he got a Nobel Prize also. DNA is proven to carry genetic information by Avery, MacLeod and McCarthy. DNA is proven to carry genetic information in the year 1944. Double helical structure of DNA was discovered by Watson and Crick in 1953. Amino acid sequence of insulin was discovered by Fred Sanger, and Fred Sanger in the year 1977 gave birth of genome sequencing also, and his sequencing method was popularly known as Sanger sequencing and enzymatic method of sequencing. He used dideoxyribonucleotides in order to cut the sequence at specific sites and after that to sequence it. Sickle cell disease occurs due to change in dash amino acid, that is only single amino acid, glutamine replaced by valine. Okay. The first complete synthesis of a gene occurred in the year 1971. Genetic engineering technique to cut and paste DNA using restriction enzyme is perfected by Stanley Cohen and Herbert Boyer. Technology to produce monoclonal antibodies is developed by Kohler and Milstein. Don't get confused between these two questions. First one is genetic engineering technique to cut and paste DNA using restriction enzyme, which was perfected by Cohen and Boyer, and technology to produce monoclonal antibodies was developed by Kohler and Milstein. Dash developed the PCR technique, that was Carey Mullis. First recombinant vaccine approved for human use is hepatitis B vaccine, that is quite dangerous than any other kind of hepatitis. Dash is the first genetically modified plant approved for the field test, that is viral resistant tomato. Initially all the experiments made on genetic modifications were on tobacco, but the first modified plant for field test was tomato. Genetically modified tomatoes are sold in USA for the first time in the year 1994. Human Genome Project was launched in 1990. The draft version of Human Genome Project was published in the year 2002. Remember, Human Genome Project was launched in 1990, and after the 12 years, the first draft version of Human Genome was published in 2002. Next, we will discuss some of the basic information of tissue culture. Callus formation in the decorticated elm tree was discovered by, options are Duhamel, Montier, Morgan, Heberland and Retchinger. Answer is callus formation in the decorticated elm tree was discovered by Duhamel, Montier. Whose work was forward for the discovery of plant tissue culture? The answer is Montier only. He was the pioneer worker in plant tissue culture. Name the scientist who expressed the view that each living cell of a multicellular organism would be capable of developing independently if provided with proper external condition was Swan. I repeat, scientist who expressed the view that each living cell of a multicellular organism would be capable of developing independently if provided with the proper external conditions, that was Swan. Swan is also known as father of cell culture. Okay. Name the scientists who proposed the polarity in development of buds from the upper portion and roots or scalus and from the lower portion of a stem piece was watching. Callus formation on isolated stem fragments and root slices is discovered by Ratchinger. I repeat, callus formation on isolation stem fragments and root slices is discovered by Ratchinger. Term totipotency was coined by totipotency. This term was coined by somebody else. Here, Morgan is not the scientist. It is given wrongly. I'll confirm and let you know. The term totipotency means capacity of a cell to form an individual or complete plant. Father of plant tissue culture is Haberland. Embryo culture was first demonstrated by Lebach. Father of micropropagation is Ball. Remember, father of micropropagation is Ball. The work on organogenesis in tobacco callus is first started by Skoog. Dash initiated studies on crown gall and tumor formation in plant, that is by White and Brown. I repeat, White and Brown initiated studies on crown gall and tumor formation in plants. The virus-free plants can be recovered from infected plants through shoot meristem culture was discovered by Morel and Martin. Nurse culture technique was established by Muir. I repeat, nurse culture technique was established by Muir. The first suspension culture by transferring callus fragments to agitated liquid medium was obtained by Hinderbrandt, Riker, and Muir. Kinetin was identified by Skoog, Miller, Saltz, and Strong. Kinetin was discovered by, that is cytokinin, which was discovered by Skoog and Miller initially, after that Saltz and Strong. Phenomena of somatic embryogenesis in suspension culture of carrot was observed by Stewart, Mapps, and Mears. The phenomena of somatic embryogenesis in suspension culture of carrot was observed by Stewart, Mapps, and Mears. First suspension culture by transferring callus fragments to agitated liquid medium was obtained by Haberland, Hinderbrandt, then Riker, and Muir. The somatic embryo formation from callus culture of carrot grown on an agitated agarified medium was observed by Reinert. Remember, somatic embryo formation from callus culture of carrot grown on an agarified medium was observed by Reinert. Technique of isolation and culture of protoplasts after digesting the cell walls enzymatically was discovered by Cocking. Planting techniques was invented by Bergman. Planting techniques were invented by Bergman. Meristem culture was discovered by Morel. Meristem culture was discovered by Morel. Then tissue culture technique was given by Guha and Maheshwari. The concept of developmental stages in culture in vitro was developed by Morashige. I repeat, the concept of development stages in cultures in vitro was developed by Morashige. Concept of development stages in cultures in vitro culture was developed by him. The polyethylene glycol method for, that is PEG method for a direct delivery of DNA into protoplast was developed by Krenz, Molondiz, and Williams. PEG-mediated direct delivery of DNA into protoplast was developed by Krenz, Molondic, and Williams. Simple method for large-scale electrofusion of protoplast was developed by Watts and King. Large-scale electrofusion of protoplast was developed by Watts and King. Basic requirements for successful genetic engineering are restriction enzymes, cloning vehicles or vectors, then detection and selection of cloned genes. Prezygotic barriers to hybridization are failure of pollen to germinate and poor pollen tube growth. Post-zygotic barriers to hybridization are lack of endosperm development. Pre-zygotic means before the formation of zygotes, that means at the gametic conditions only, and post-zygotic means after the formation of zygote, that may be endosperm development or embryo development, etc. Pre-zygotic barriers to hybridization can be overcome by in vitro fertilization. There are 4 options given: in vitro fertilization, embryo culture, ovule culture and pod culture, but pre-zygotic barriers to hybridization can be overcome by in vitro fertilization. If it is post-zygotic means we may go for embryo culture, pod culture, ovule culture, etc. Ovary culture also. Embryo culture has been successful in overcoming the problems like low seed set, seed dormancy, slow seed germination. Delagold and AC Chang are the commercial varieties of tobacco produced by protoplast fusion. This you need to remember, often you asked in many of the competitive exams. Delagold and AC Chang are the commercial varieties of tobacco and these are produced by protoplast fusion. Production of novel hybrids through protoplast fusion should focus on agriculturally important traits, means not on all the traits, whichever are very important, those kind of things. Then somatic hybrids integrated into a conventional breeding program. Then extension of protoplast regeneration to a wider range of crop species. Somatic हाइब्रिड्स क्या होते हैं जिसको नॉट द जर्म सेल्स बट सोमैटिक सेल्स से लिया जाता है, दैट मीन्स अपार्ट फ्रॉम द रिप्रोडक्टिव सेल्स. एंड एक्सटेंशन ऑफ प्रोटोप्लास्ट रीजेनरेशन. प्रोटोप्लास्ट मीन्स सेल विथआउट सेल वॉल, तो सेल वॉल रिमूव करके प्रोटोप्लास्ट जो बनाते हैं उसको रीजेनरेशन करके वाइड रेंज ऑफ क्रॉप स्पीशीज में यूज़ कर सकते हैं, मतलब उसको बहुत सारे क्रॉप स्पीशीज से एक्सट्रैक्ट कर सकते हैं. In vitro method used to generate haploids is culture of excised ovaries and ovule, bulbosum technique of embryo culture and culture of excised anthers and pollen. The question is, in vitro method used to generate haploids is for culture of excised ovaries and ovules, bulbosum technique of embryo culture and culture of excised anthers and pollen. Method used for in vitro propagation are enhancing axillary bud breaking, then production of adventitious buds and somatic embryogenesis. Next we shall move to the cell biology questions. Cell differentiation in animals is irreversible, means once differentiated again you cannot take out a hand of any person and multiply it, okay, but in case of plants you can just break a branch and propagate further. Not in all cases but in many of the plant species, especially in the angiosperms. Cell differentiation in plants is reversible, okay. The inherent potentiality of a plant cell to give rise to a whole plant is described as morphogenesis, but it is basically defined as totipotency, that is inherent potentiality of a plant cell to give rise to a whole plant is cellular totipotency. For a differentiated cell to express its totipotency, it should undergo differentiation followed by dedifferentiation and redifferentiation. डिफ्रेंशिएशन होता है सिंगल सेल डिफ्रेंशिएट होकर मैनी सेल्स फॉर्म हो जाएगा, and de-differentiation होगा that many cell to a, that means the differentiated cell will give rise to again single cell, that is differentiation. Redifferentiation मतलब again it will undergo, after de-differentiation redifferentiation will be occurring. The phenomena of a mature cell reverting to the meristematic state and forming undifferentiated callus tissue is termed as de-differentiation. Remember यहाँ पे आपको differentiation, de-differentiation and redifferentiation का definition याद रहना बहुत ही ज़रूरी है, okay. See, what is de-differentiation? That is the phenomena of a mature cell reverting to the meristematic stage. Meristematic मतलब divide हो जाएगा. Mature cell meristematic मतलब dividing form में उस टाइप ऑफ सेल को ये देगा, जन्म देगा, एंड फॉर्मिंग अनडिफरेंशिएटेड कैलस टिश्यू। रीडिफ्रेंशिएशन क्या होता है? फिनोमिना ऑफ कन्वर्शन ऑफ द कंपोनेंट सेल्स ऑफ कैलस टिश्यू टू होल प्लांट और ऑर्गन। रीडिफ्रेंशिएशन इज नथिंग बट इट इज ए सेकेंड स्टेज ऑफ डिफ्रेंशिएशन। ठीक है, यहाँ पे होता है कन्वर्शन ऑफ कंपोनेंट सेल्स ऑफ कैलस टिश्यू टू होल प्लांट, मतलब डिफ्रेंशिएटिंग एक सेल होता है वो होल प्लांट में कन्वर्ट हो जाएगा। वही डी डिफ्रेंशिएशन में क्या हो जाएगा? मैच्योर सेल मेरिस्टेमेटिक हो जाएगा। The phenomena of totipotency is demonstrated with the experimental involving carrot. टोटिपोटेंसी को कैरेट में फर्स्ट टाइम स्टडी किया था। द फेनोमिना ऑफ मॉर्फोजेनेसिस डिपेंड्स ऑन द फैक्टर्स लाइक पोलैरिटी, देन डिफरेंशिएशन, regeneration of individual cells. Dash concluded that organ building in the fate of the cell is function of its position watching. Under in vitro condition, this can be achieved by de novo origin of organs and de novo origin of embryos. Organogenesis means genesis of organs like shoots, roots, leaves and flowers. Earliest report on induction of shoot organogenesis in vitro was by using white using tobacco hybrid. मतलब white ने earliest report on induction of shoot organogenesis in vitro tobacco में report किया था. First observation of root formation was reported by Nabokov using carrot callus. The regulatory mechanism, that is balance between auxin and cytokinin, is recognized by Nabokov and Skoog and Miller. The answer here is Skoog and Miller. The relatively high level of auxin to cytokinin favored high level of auxin to cytokinin. Auxin जहाँ ज़्यादा होता है that will give rise to roots. Then where cytokinin is more, it will give rise to shoots. And if the amount is equal, that gives rise to callus. Relatively high level of cytokinin to auxin is favored during shoot formation. Okay. The genesis of shoots from the explants or calli is termed as callogenesis. Genesis of root from the explant or calli is termed as rhizogenesis. Callogenesis, calli formation होना and root formation होना है तो rhizogenesis. Event of organogenesis is discovered by Terry. Remember, event of organogenesis was discovered by Torrey. The somatic embryogenesis was first discovered by Stewart. Various theories of somatic embryogenesis are cell isolation theory, differentiation theory, then intercellular communication and cytodifferentiation theory. Remember, various theories of somatic embryogenesis are cell isolation theory, differentiation theory, intercellular communication and cytodifferentiation theory. Cell isolation theory was proposed by Stewart. Differentiation theory was proposed by Halperin. Then intercellular communication and cytodifferentiation theory was developed by Street. Three things you need to remember. Cell isolation theory proposed by Stewart, differentiation theory by Halperin, and intercellular communication and cytodifferentiation theory was proposed by Street. And these three theories are the theories of somatic embryogenesis. The explant physiology and culture environment theory was proposed by Street only. The predetermination theory was proposed by Tisserand. Members of Umbelliferaceae family readily form somatic embryos in culture. That means in Umbelliferaceae family, somatic embryo induction is quite easy. High concentration of dash favors shoot initiation, that is nothing but cytokinin or kinetin. High concentration of dash favors rooting, auxin. In somatic embryogenesis, dash is required for induction of embryonic cells and maintenance of proliferative growth, that is auxin is required in somatic embryogenesis for induction of embryonic cells and maintenance of proliferative growth. Embryo formation can be induced by transferring the callus to a medium lacking auxin. Dash is used most successfully to obtain rapid growth of somatic embryo into plant is gibberellic acid. That is, gibberellic acid is used most successfully to obtain rapid growth of somatic embryos into plants. Mineral solution widely used by early investigators was NOPS medium. Remember, mineral solution widely used by early investigator was NOPS solution. The medium in tissue culture should provide nutrients for the survival of the plant cells, tissues, organs under culture and optimal physical conditions of pH, osmotic pressure, etc. Next question is, the callus culture was first developed by Gotthardt. Systematic study of mineral requirement of plant tissue and organs in culture was made by Murishige and Skog, Vasil and Hildebrand, Nish and Nish. The mineral nutrient that plays a vital role in growth and differentiation of culture tissue is nitrogen. Remember, nitrogen plays very crucial role in growth and differentiation of cultured tissues. Nitrogen generally supplied in NH4, that is ammonical form. The range of inorganic nitrogen varies between 25 mm to 60 mm in tissue culture medium. This you need to remember very carefully. Inorganic nitrogen content supplemented to tissue culture medium is 25 mm to 60 mm. Phosphorus level greater than dash are inhibitory to growth of tissue is 2 mm. Means you should not provide more than 2 mm phosphorus to the growing tissue culture media. Then the optimum concentration of K, that is potassium, needed is 20 mm. That is more than phosphorus. K needed is 20 mm. At low nitrogen concentration, presence of potassium element enhances the formation of somatic embryos. That means if nitrogen content is less, also potassium manages this. Sulfur is provided in the form of sulfates. The standard carbon source is 2 to 5% sucrose. The standard carbon source is sucrose. Then universally required vitamin in tissue culture medium is thiamine HCl. Remember, standard carbon source after sucrose is glucose only. Glucose is readily available, but sucrose need to be converted. Okay. Universally required vitamin in tissue culture medium is thiamine HCl. Vitamins used in tissue culture are calcium, pantothenate, then pyridoxine and nicotinic acid. Commonly used auxins in tissue culture are IAA, IPA, NAA and 2,4-D. The commonly used cytokinin are BAP, 2IP, kinetin and zeatin. Auxin are diluted in ethanol and dilute NaOH. Cytokinins are diluted in dilute HCl and dilute NaOH. In tissue culture, auxins have been used for cell division and root differentiation. Remember, this is very important. In tissue culture, auxins have been used for cell division and root differentiation. In tissue culture, cytokinins are used for differentiation of adventitious shoots from callus and organs and shoot proliferation. ऑक्सिन का फंक्शन क्या होता है? सेल डिवीजन और रूट डिफरेंशिएशन. लेकिन साइटोकाइनिन क्या करता है? डिफरेंशिएशन ऑफ एडवेंटिशियस शूट्स फ्रॉम कैलस एंड ऑर्गन्स एंड शूट प्रोलिफरेशन. In tissue culture, gibberellins have been used for normal development of plantlets from in vitro formed adventitious embryos. Gibberellins कहाँ यूज़ करते हैं? Normal development of plantlets from in vitro formed adventitious embryo. High auxin, low cytokinin ratio favors root formation, embryogenesis and callus formation. Low auxin to high cytokinin क्या करता है? एडवेंटिशियस रूट एंड ऑक्सिलरी शूट्स का फॉर्मेशन करता है. द ऑक्सिन साइटोकाइनिन रेशियो इज एसेंशियल फॉर initiating root formation, embryogenesis, callus formation, then induce formation of adventitious or axillary shoots, then chloroplast formation and other processes. Naturally occurring gibberellin involve internode elongation, enhancement of flowers, fruits and leaf size, then germination and vernalization. जिबरेलिन्स आर सोल्यूबल इन कोल्ड वाटर. ठीक है, जिबरेलिन्स कोल्ड वाटर में सोल्यूबल है. इथाइलिन इज इन्वॉल्व इन फ्रूट मैचुरेशन, एब्सिशन, सेनेसेंस. मतलब ये एजिंग हार्मोन होता है इथाइलिन. एब्सिसिक एसिड इज इन्वॉल्व इन एनफोर्सिंग डॉर्मेंसी, एब्सिशन, रेगुलेटिंग अर्ली स्टेजेस ऑफ एम्ब्रियो डेवलपमेंट. मतलब एब्सिसिक एसिड क्या करता है, अर्ली स्टेज में एम्ब्रियो डेवलप करने में हेल्प करता है, फिर डॉर्मेंसी एनफोर्स करेगा एंड एब्सिशन लाएगा. एब्सिसिक एसिड इज रिक्वायर्ड फॉर नॉर्मल ग्रोथ, प्रमोट्स मॉर्फोजेनेसिस एंड डेवलपमेंट ऑफ सोमैटिक एम्ब्रियोज. डैश प्रमोट्स इथाइलिन बायोसिंथेसिस एंड एपिनेस्टी. ब्रासिनोस्टेरॉइड्स एक काइंड ऑफ हार्मोन्स है जो प्रमोट करता है इथाइलिन बायोसिंथेसिस एंड एपिनेस्टी. जास्मोनिक एसिड इज ए मिथाइल एस्टर. Remember, jasmonic acid is a methyl ester. Optimum pH for tissue culture is 5.8. The neutral pH is 7, लेकिन टिश्यू कल्चर के लिए 5.8 चाहिए. The substitutes for agar are gelatin, silica gel and acrylamide gel. हम सिर्फ अगार ही टिश्यू कल्चर में यूज़ करने की कोई ज़रूरत नहीं है. उसके साथ साथ में जिलेटिन, सिलिका जेल, अक्राइल अमाइन जेल भी यूज़ कर सकते हैं. लेकिन अगार काफ़ी अफोर्डेबल भी है एंड दिस गिव्स बेटर न्यूट्रिशन और बेटर मीडिया फॉर द ग्रोविंग टिश्यूज़. मतलब मॉइस्चर काफ़ी हद तक ये होल्ड करके रखेगा. The osmoticums used in tissue culture medium are sorbitol, mannitol, sucrose, fructose, galactose. ये सब क्या है, osmoticum. Then the process by which meristematic cells are converted into two or more types of cells, tissues or organs which are qualitatively different from each other is differentiation. मैंने डी डिफ्रेंशिएशन एंड रीडिफ्रेंशिएशन के बारे में आपको बताया था. तो डिफ्रेंशिएशन का क्या ये होता है डेफिनेशन. मेरिस्टेमेटिक सेल्स कन्वर्ट होंगे टू और मोर टाइप ऑफ सेल्स में, टिश्यू में एंड ऑर्गन में. ठीक है, मेरिस्टेमेटिक सेल टिश्यू ऑर्गन में कन्वर्ट हो रहा है. डी डिफ्रेंशिएटेड सेल्स में क्या हो रहा है, मेरिस्टेमेटिक सेल्स से डिफ्रेंशिएशन में शुरू किया था, फिर डिफरेंट टाइप्स ऑफ सेल्स बन गए एंड ऑर्गन्स बन गए. लेकिन डी डिफ्रेंशिएशन में क्या हुआ, अनऑर्गेनाइज्ड टिश्यू यहाँ पे ऑर्गेनाइज्ड टिश्यू में कन्वर्ट हो गया, मतलब मेरिस्टेमेटिक बन गया. देन डी डिफ्रेंशिएशन अकरिंग इन एन अनडिफ्रेंशिएटेड टिश्यू प्रोसेस क्या होता है, उसको रीडिफ्रेंशिएशन बोलते हैं. रिमेंबर, de-differentiation occur in an undifferentiated tissue is redifferentiation. एक और डेफिनेशन है, जेनेसिस ऑफ एन एंटायर प्लांट फ्रॉम कल्चर्ड एक्सप्लांट्स डायरेक्टली और वाया कैलस indirectly is called as regeneration. Genesis of an entire plant from cultured explants directly or via callus indirectly. Directly कैसे होगा, by कल्चर्ड एक्सप्लांट से, और इनडायरेक्टली कैसे होगा, कैलस फॉर्मेशन से. तो उसको बोलते हैं रीजनरेशन. अटेनमेंट ऑफ बायोलॉजिकल ऑर्गेनाइजेशन और फॉर्म इज टर्म्ड एज मॉर्फोजेनेसिस. यहाँ पे बहुत सारे मैंने ऑप्शन डेफिनेशंस के बारे में डिस्कस किया, जैसे कि डिफरेंशिएशन, डी डिफरेंशिएशन, रीडिफरेंशिएशन, फिर रीजनरेशन, फिर मॉर्फोजेनेसिस. ये इतने भी डेफिनेशन बहुत ही ज़रूरी है. अब देखते हैं मॉर्फोजेनेसिस क्या होता है. वो होता है अटेनमेंट ऑफ बायोलॉजिकल ऑर्गेनाइजेशन और फॉर्म इज मॉर्फोजेनेसिस दैट कैन बी अचीव्ड बाय डी नेवो ओरिजिन ऑफ ऑर्गन्स और डी नेवो ओरिजिन ऑफ एम्ब्रियोज. डी नेवो मतलब आर्टिफिशियल कंडीशन में. द अर्लीस्ट रिपोर्ट ऑन इंडक्शन ऑफ शूट ऑर्गेनोजेनेसिस इन विट्रो वॉज वाइट ने रिपोर्ट किया था शूट रीजन ऑर्गेनोजेनेसिस इन विट्रो. अर्लीस्ट रिपोर्ट ऑन इंडक्शन ऑफ रूट ऑर्गेनोजेनेसिस वाज नोबेकोर्ट. इंडक्शन ऑफ शूट ऑर्गेनोजेनेसिस इन विट्रो वाज अचीव्ड प्राइमरीली इन टोबैको. Remember, induction of shoot organogenesis in vitro was achieved primarily in tobacco. Induction of root organogenesis in vitro was achieved primarily in कैरेट। शूट की बात आते हैं ऑर्गेनोजेनेसिस इन विट्रो, तो वो टोबैको में था, एंड रूट का होता है तो वो कैरेट में था। ओके, डेवलपमेंटल सीक्वेंस इन्वॉल्विंग एन इंटरवीनिंग कैलस स्टेज इज टर्म्ड एज इनडायरेक्ट ऑर्गेनोजेनेसिस। इंटरवीनिंग कैलस मतलब जहाँ पे कैलस आ जाएगा डेवलपमेंटल सीक्वेंस में, वो उसको इनडायरेक्ट ऑर्गेनोजेनेसिस बोलते हैं। अगर कैलस इन्वॉल्व नहीं है तो वो होगा डायरेक्ट ऑर्गेनोजेनेसिस। Abnormal structures developed during organogenesis is called organoid. Abnormal structures developed during organogenesis are called organoids. देखिए, जहाँ भी अनफेवरेबल कंडीशन होते हैं या नेगेटिव टर्म्स में हम बात करते हैं तो नॉइड लगाते हैं। ओके, localized meristematic cells on a callus which give rise to shoot or roots are termed as meristemoids. Localized meristematic cells or callus which gives rise to shoot or roots are termed as meristemoids. Seed culture is used to increase the efficiency of germination of seeds. Precocious germination by application of plant growth regulators or production of clean seedlings for explants or meristematic culture. तो एम्ब्रियो कल्चर क्यों यूज़ करते हैं? टू ओवरकम एम्ब्रियो एबॉर्शन ड्यू टू incompatibility barriers, to overcome seed dormancy and self-sterility of seeds, then embryo rescue in distant hybridization. ओव्यूल और ओवरी कल्चर कहाँ यूज़ कर सकते हैं? प्रोडक्शन ऑफ हैप्लॉइड में, इनिशिएशन ऑफ सोमैटिक एम्ब्रियोनिक कल्चर में, टू ओवरकम एबॉर्शन ऑफ एम्ब्रियोस ऑफ वाइड हाइब्रिड्स. Multiplication of genetically identical copies of a cultivar by asexual reproduction is called as clonal propagation. Clone क्या होते हैं? These are the n number of explants generated by asexual propagation. In nature, clonal propagation occurs by vegetative propagation or apomixis. Apomixis में क्या होता है? Here, seed development without meiosis and fertilization, or embryo development without fertilization. Vegetative propagation is by regeneration of new plants from vegetative parts. Advantage of tissue culture: production of large number of true to type plantlets from a single plant, less time requirement, less space requirement. मतलब जब भी हम ग्रीन हाउस कंडीशन या कंट्रोल्ड एनवायरनमेंट में करते हैं तो नंबर ऑफ सीजन्स हम बढ़ा सकते हैं. मतलब इट इज़ नॉट entirely dependent on the environment but on the controlled condition and controlled supplement of nutrients as well as water, we can adjust the growth periods also and multiply on a large scale. Total number of stages in micropropagation is 5. Dash stage involves the propagation or mother plant. Stage 0. मदर प्लांट को जब कट करेंगे और उसको सेलेक्ट करेंगे, ये सब स्टेज 0 में आएगा. डैश स्टेज इन्वॉल्व प्रिपरेशन ऑफ मदर प्लांट, वो स्टेज 0 है. प्रिपरेटिव स्टेज को स्टेज 0 बोलते हैं. स्टेज 1 क्या होगा? इनिशिएशन ऑफ कल्चर. स्टेज 2 होगा मल्टीप्लिकेशन. स्टेज 3 होगा रूटिंग ऑफ शूट्स. स्टेज 3 में ही ट्रांसप्लांटिंग करते हैं. फिर इसमें सुटेबल एक्सप्लांट्स फॉर प्रोड्यूसिंग वायरल फ्री प्लांट्स इज शूट टिप. द ऑक्सीडेशन ऑफ फिनोलिक, तो देखिए पहले रूटिंग करते हैं स्टेज 3 में, फिर पहले रूटिंग होता है, फिर शूटिंग होगा, देन प्लांट्स ट्रांसफ़र करेंगे कि आर्टिफिशियल मीडिया में, तो वो ग्रो हो जाएगा. देन उसको बाहर मतलब एक्लेमेटाइज करने के लिए या हार्डनिंग के लिए लैब में या वी कैन से ग्रीन हाउस में ट्रांसफ़र करेंगे. वो क्या होगा? स्टेज 5. तो 5 स्टेजेस है, स्टेज 0 से लेके स्टेज 5 तक. ओके, सुटेबल एक्सप्लांट फॉर प्रोड्यूसिंग वायरस फ्री प्लांट इज शूट टिप. और मेरिस्टेमेटिक जो पोर्शन होता है उससे हम बहुत अच्छे तरीके से एक्सप्लांट ले सकते हैं which are virus free. The oxidation of phenolic compounds leached out from the cut surface of explant in tissue culture leads to browning of the medium. Here you need to remember the oxidation phenolic compounds leached out from the cut surface of the explant in tissue culture leads to browning of the medium. Even आपने देखा होगा जब एप्पल कट करते हैं तो वो ब्राउन कलर में, या whenever we cut the banana, we get the brownish appearance or dull appearance of the fruit. That is because of the leaching out of phenolic compounds. The oxidation of phenolic compounds leached out from the cut surface of eggplant leads to browning. Browning of the medium is common problem of adult tissues from woody species. मतलब यंग टिश्यू जितना लेंगे या मेरिस्टेमेटिक टिश्यू जितना लेंगे वहाँ पे हम ब्राउनिंग को अवॉइड कर सकते हैं. क्रूशियल स्टेज इन टिश्यू कल्चर इज मल्टीप्लिकेशन. इन टिश्यू कल्चर मल्टीप्लिकेशन इज थ्रू कैल्यूजिंग, adventitious bud formation and enhanced axillary branches. Remember, in tissue culture multiplication is through callusing, adventitious bud formation and enhanced axillary branching, whereas multiplication is very crucial step in plant tissue culture. Friends, the information furnished by me, I think it is helpful for you. If you like this video, please do not forget to share, like and keep on subscribing. Thank you once again.